Yeah, a Mexican approach in Slytherin cancer with nanotechnology and snake venoms. So the Mexican approach is just like, because we do it in Mexico, there's not so much a difference per se regarding what other laboratories or research groups are doing in other parts of the world. So I do like to divide my presentations generally in three parts. So first of all, I will be telling you just a little bit why is it that we study snake venoms with cancer and using nanotechnology. How is it that we actually perform such studies, and some of the results that we got from my PhD project. So starting with the why is it that we study cancer? So when we consider the cancer panorama in Mexico, we can see that the most incident cancer types are prostate, breast, and uterine cancers. Specifically, breast cancer takes the second place. When we see the mortality rates from these cancer types, we see that breast cancer, it's also the second place. And nevertheless, when we see the Mexican panorama of prevalence of cancer, we actually see that there's a slight shift, and breast cancer takes the first place. Hence, we can consider breast cancer in Mexico a public safety, not public safety, sorry, a public health issue. And why is it? Because, well, breast cancer affects mainly women, of course, and culturally in Mexico, I don't know if in other parts of the world, but at least in Mexico, if you ever feel bad, Mexicans don't tend to go to the doctor or to a hospital because we're pretty much afraid of doctors and hospitals. So why is it that we believe that breast cancer it has such a high prevalence within the country? Well, it's pretty much because people don't like to go and check themselves. And nevertheless, that doesn't take out the issue that breast cancer is a public health issue within the country. So having said that, we can establish that we have a health issue that we can actually study and try to either resolve or pretty much help resolve in a future. Now, how is it that we're actually trying to help resolve this issue? Well, with the use of nanotechnology, considering that breast cancer is of importance worldwide, and that most of cancer types they are rather difficult to treat, depending, of course, on the stage that it is currently developing or that it was found out. Now, nanomedicine, which is pretty much the use of nanotechnological concepts or techniques alongside novel medicines, pharmaceuticals, or bioactive components, by combining these two, we can actually help with the development of either novel pharmaceuticals or new techniques to either detect cancer on earlier stages or to treat cancer on later stages. And how is it that we actually do that? Or how is it that it actually works? So I work with polymetic nanoparticles, which you can consider them as super small balls of polymers. You can pick any polymer of your choosing. I usually work with PLGA, which is an FDA approved material to develop drug delivery systems. Now, PLGA specifically, it's composed of lactic acid and glycolic acid, which both are found within the Krebs cycle. So it's a biocompatible biosynthetic material. Now, what is it that we actually do? So we use polymeric nanoparticles, and we can consider that we have an API or an active pharmaceutical ingredient or any peptide, any protein that can be found within an animal venom. Let's say that's the disulfide-rich protein from spiders, for example. Now, what is it that we actually do? Well, what we try to mainly do is to encapsulate these APIs or these bioactive components within the nanoparticle. Now, why do we try to encapsulate them? Well, first of all, because we can avoid a rapid degradation from these pharmaceutical ingredients or peptides or proteins. So if any of you have ever worked with peptides or proteins within either cells or in vivo systems, you will most likely have noticed that they have a super rapid degradation rate. And well, that's one thing that we can actually do, avoid the rapid degradation rate, because the first thing that our system or an in vivo system will recognize, it's the particle itself and not the component, the bioactive component. Now, another advantage is that we can actually present a sustained release profile from the nanoparticle. This means that we can actually measure and control how often will this bioactive component will be released from the nanoparticle matrix. And this has a lot of advantages. So imagine that instead of getting a chemotherapeutic cycle every 2 or 3 weeks, you can actually prolong it for every month, month and a half, or every 2 months. So that way, from one simple dose or one simple dosage, you can actually retain or maintain a therapeutic level in your plasma blood levels of the either bioactive component or the pharmaceutical ingredient, which means also paying less and having a more patient compliance with the therapeutical cycle. And lastly, we can actually passively or actively target a drug delivery. So this is a super interesting thing that happens with nanoparticles. And passively, some solid tumor cells, they are more permeated with nanoparticles, which tends to accumulate nanoparticles within tumor cells. So what happens if a tumor cell it's way more permeated towards nanoparticles, they will most likely be retained in a longer time, in a longer lifespan within the tumor cell. Now, the tumor cell, what it will actually eventually do, it's to, let's say, cut off the nanoparticle matrix. It will pretty much dissociate it. And what will happen then? Well, everything that was encapsulated within these nanoparticles, it can also happen with microparticles. It will release either the bioactive component or the active pharmaceutical ingredient within the tumor cell. Hence, we will have like a passively target drug delivery system. We can actually do it in an active manner by putting some antibodies or specific antigens within the surface of the nanoparticle. And in that way, we can actually actively tell the nanoparticle to concentrate on a specific target. But passively, it's been reported that this can happen by just having a nanoparticle size of over 400 nanometers or less. So that's pretty much what we do. Now, why is it that we actually do it with snake venoms? So here's Mexico, the political map of Mexico, and just some quick data. So we're the country with the highest diversity of overall snakes, either considering venomous and non-venomous snakes in the world, with over 448 species. And in the state of Sonora, which is highlighted right here, when we have got the Sonoran Desert, the most biodiverse desert within the world, we have more than 15 highly venomous species in our state. And what's like interesting regarding the state of Sonora? So we got two families of snakes which are considered as highly venomous. So we got the viper family or the Viperide family, where we have over 14 highly venomous species, considering some snakes whose local names are Pichicuates from the genus Achistrodon, and of course also the Curtalus snakes or rattlesnakes. And we got 3 species of elapids, 2 coral snakes, and the Hydrophis platurus sea snake, or the yellow-bellied sea snake, which happens or occurs alongside the Sonoran coast. So we can consider that within the state of Sonora, which is the second largest state in Mexico, we have a rather high diversity of highly venomous snakes. And what's important regarding having like a high diversity of venomous snakes? So just like Naim presented in his presentation, snake venoms are, well, they are highly diverse, and they got some toxin families which are super interesting for pharmaceutical research and development. And you may very well be asking yourselves, why is it that they're interesting? Well, if we consider the anti-tumoral potential that some of these toxins have, just to give you a heads up, PLA2 are phospholipases or A2 phospholipases. The orange ones are snake venom metalloproteases, green ones are snake venom serine proteases, and other toxin families. So it's been reported that most, if not all, of these toxin families can have some anti-tumoral or anti-cancer potential up to a certain degree, either by inhibiting angiogenesis, which will prevent metastasis, or it will prevent the initiation of a metastasis cascade. Metalloproteases are rather good at inhibiting cell adhesion and the initiation of a metastasis cascade. L-amino acid oxidases, they tend to concentrate reactive oxygen species on the membranes, hence inducing apoptosis on tumoral cells, and so on. So there's like a lot of things that we can actually do with these snake toxins. Now, considering all of the aforementioned, if we have a public health issue regarding breast cancer, we have a rather interesting way to deliver these bioactive systems, which is using nanotechnology or polymeric nanoparticles. And we have some rather interesting bioactive components, which are snake venom toxins. If we combine all of these, well, we can actually like visualize where I'm going. We're trying to encapsulate all of these or isolated toxins in order to deliver them to tumoral cells, and we'll evaluate how good they are at actually inhibiting the growth of cancer cells. Now, how is it that we actually do it? So this is probably super common in our network, which is the extraction of the venom, lyophilizing. Oh, sorry, I didn't change it from Spanish to English, but lyophilizing the venom in order to perform characterization studies or analysis, checking the venom migration in an SDS page, gel, identifying the venom proteins, evaluating its hemolytic activity and its cytotoxicity within the cancer cells or normal non-transformed cells, and all of that stuff. How is it that we actually evaluate it? Well, we pretty much evaluated only in vitro. So we did it by culturing mammalian cancer cells and using an MTT cell viability assay. Well, we pretty much just incubated cancer cells up to a certain degree, and then we put some venom and venom nanoparticles, and we quantified how much of the cells were dead after a certain period of time, which was 24 hours. This is rather biophysics, so I don't know if you guys are interested, but how is it that we actually encapsulate the venom? So in my case, I used PLGA, polylactic-glycolic acid. It's the material that I told you earlier. Now, this material is, interestingly, it's not hydrophilic, unlike proteins. So how is it that we actually encapsulate a hydrophilic component into a hydrophobic component? So we do it by a double emulsion process. So we pretty much just sonicate the materials. This gives us like a nanoparticle that has an organic solvent, which is encapsulating uh, a hydrophilic component. And after this process, we actually put some aqueous solution, we sonicate it again, therefore we got a hydrophilic nanoparticle encapsulating some organic solvent and also the proteins. We just let the solvent evaporate itself, and at the end we get a hydrophilic nanoparticle stabilized by some surfactants, which is PBA, polyvinylic alcohol, which is also used in a lot of 3D printing It's a different type of PVA, but it's pretty much the same material. So these are biophysics. I don't know if you guys are interested, but pretty much we sonicate everything through a double emulsion process. And this gives us some nanoparticles, which are super effective to encapsulate hydrophilic components. Now, going for the interesting part, which are the results that we got. So first of all, we characterized the venom, and we noticed that the venom of this specific rattlesnake that we use, Crotalus molossus, which is also known as the blacktail or northern black-tailed rattlesnake, it's highly diverse. So we can't actually say that it's highly diverse just from an SDS page, gel electrophoresis. We can pretty much like see some gel bands that are among the 66 kilodaltons range. We got some other small, almost phantom bands on the 45 range, on the 20s range, and on the 14 range. Now, I didn't put the table in here, but we actually did an LC-MS analysis, and we were able to identify each of the proteins that were composing the black-tailed rattlesnake venom. So extrapolating that information, the theoretical mass from each of the proteins, we were actually able to determine that each of these bands belonged to some metalloproteases, some amino acid oxidases, some serine proteases, phospholipases, and C-type lectins, which led us to develop this super small pie chart of how the venom diversity of this rattlesnake is composed. And you can see in here that it's mainly composed of phospholipases, metalloproteases, and on a lesser way from C-type lectins, amino acid oxidases, serine proteases, and other proteins, which are composed of like a super variable proteins, which I didn't wanted to put like so many tags in there. So I just labeled them as others. Now, we know what the venom composition of this rattlesnake is. We eventually did the cytotoxicity analysis. On the x-axis, where we can see the concentration, and on the y-axis, the cell viability of this specific type of breast cancer cell line. This is called T47D cell line, which is an hormone positive type of breast cancer cell. So what we found out is that snake venom by itself, it's rather cytotoxic to this cell line, which is something that we were expecting, right? I mean, if we see the clinical symptoms that are derived from a rattlesnake bite, we can actually infer that it would actually be highly cytotoxic to pretty much most of cells. Now, what we did later is that we actually lowered the concentration to 10 micrograms per milliliter down to 0.08 micrograms per milliliter in order to determine the IC50. So what we found out is that the IC50 of the whole venom extract of Crotalus molossus, it's 1.5 micrograms per milliliter. If you would like to consider or to compare this IC50 like to other used pharmaceutical ingredients, well, we can actually compare it to doxorubicin or paclitaxel, which are some FDA-approved chemotherapeutics, and they go around 1 microgram per milliliter, depending on the author, right? So we can actually say that the whole venom extract is, well, highly cytotoxic. We did some later on evaluations, like we evaluated the hemolytic activity, and we did some other stuff, but I just want to go like to the grain. So we actually synthesized these nanoparticles, and we were able to obtain nanoparticles ranging around 300 nanometers. So depending on the author, some authors are going to be like extremists as to say, or purists, and they will tell you that nanoparticles are particles that go from 100 nanometers and below in diameter size. Nevertheless, when working with polymers, it's rather hard to have sub 100 nanometer nanoparticles. And a 300 nanometer nanoparticle is actually pretty good for drug delivery systems and to obtain a sustained release with these bioactive components. Now, if you are asking yourselves right now, like, what's the importance of the size and what's like the good thing to see in here? So a cool thing about our nanoparticles is that they have a spherical shape and they are smooth on the surface. This actually enhances the probability that these nanoparticles will enter rather easily into the tumor cells, and they will be retained within the tumor cell. And we had an efficiency, an encapsulation efficiency of around 70%, 75%, give or take, and a loading efficiency, which this tells us how much of the nanoparticle weight is actually the protein, of around 10%, which are rather good values in this case. So we later extracted the venom from the nanoparticles, and we saw the migration pattern on an SDS gel page. And we noticed that we were extracting some metalloproteases, amino acid oxidases, phospholipases, and C-tablectins. So this tells us that at least from the toxins that we could extract from the nanoparticles, any type of cytotoxic activity that we would be seeing once we evaluated these nanoparticles, we could actually infer that they will belong or they are going to be thanks to these specific toxin families in our case. By evaluating the sustained release, we actually evaluated up to 300 hours. And this is something rather interesting. So depending on the drug delivery system that you're studying or using, you can actually get like a release of around 50 to 40% of your bioactive component on the first 24 hours. So with our system, at the first 24 hours, which would be around here, we were actually releasing only about 15% of the whole encapsulated venom. So this tells us some rather interesting things. It tells us that our nanoparticle encapsulation system actually allows us to have a sustained release of a rather dangerous bioactive components, which are the snake toxins, for a rather large period of time. Why is it interesting? Because we could actually manage, we can actually control the sustained release by modifying the encapsulation process and modifying the surface of the nanoparticle and doing other stuff. But by controlling the sustained release and having an actual sustained relief, a long sustained relief, we can actually think that it will be rather safe to put these nanoparticles within organisms, let's say mice later on, or eventually humans, because they won't be releasing all of the toxin families at the same times and with a burst-like effect, meaning that they will be releasing these venoms or these toxin families on a sustained release, which would be rather safe. When we compared the cytotoxicity of the venom by itself on the gray bars and with the venom nanoparticles, we can actually see that at higher doses they actually they are quite they resemble each other. Nevertheless, on the lower doses, we can actually see that the venom nanoparticles didn't have quite an acute cytotoxic effect on this tumoral cell line. Now we can actually infer this considering the sustained release. There will be some intra and intermolecular interactions with the polymetic matrix and the venom toxins that most likely are not allowing these venoms to be released from the nanoparticle within the cell. So this is to be expected. It can be modified for in order to have a higher cytotoxicity. Of course it can. And we did the same with lower doses, and we were able to determine the IC50 for the venom nanoparticles at 22 micrograms per milliliter. Now, according to some federal Mexican agencies, if you have a bioactive component that has an IC50 of 30 micrograms per milliliter or less, it's considered to be of importance or interesting to be continued starting in anti-cancer activity. So we pretty much just like managed to get into that range. Again, these are just preliminary analysis, since it's kind of weird to use nanoparticle and snake venoms. There are rather not so much research groups in the world that are using it right now. So at least for us, they were some like super interesting stuff. So I actually didn't mean to put this slide right here, so I'm just gonna take it out. So in conclusion, we were able to determine the venom composition of these rattlesnake venom from this rattlesnake species in northern Mexico, which is a rather the word is not unstudied, but a rather not so studied rattlesnake within the country. We were able to synthesize and characterize these nanoparticles, and we were able to determine that it was a rather good system to obtain a sustained release and to keep having a cytotoxic activity after the encapsulation process. And that's pretty much it. I'm sorry if I took longer than expected. This is the medical biophysics lab members, some of the members, and which is a highly diverse laboratory here in Mexico. We got some electrical engineers and geologists and every single type of bachelor degree that you can imagine. And that's pretty much it. So thank you so much, everyone. In case you need any information or you're interested, here's my email. Feel free to send me a message, and don't be shy. If you want to send me a WhatsApp message or anything, this is my phone number. And that's pretty much it for my part. Thank you so much.
Thank you very much for the talk. And you were just a little bit over time.
I'm sorry.
Are there any questions? Else I have always questions about snake venoms. You showed the SDS gels with the venom that was incorporated into the nanoparticles. These were reduced gels, right?
Yeah, I forgot to tell you guys that. Yeah, these were reduced gels under denaturalizing conditions.
But did you check also the non-reduced gels?
We didn't. So one of the main issues that we have right now doing research in Mexico, I don't want to enter into any political things or anything, but we have been working under some serious national budget cuts for research. So considering that COVID had something to do within our time doing the PhD, and considering the budget cuts, we weren't able to do as many studies as we wanted to. So yeah, we weren't able to. It's not that we weren't able to. We didn't have the budget to do more studies.
Because it would be interesting to see if the C-type lectins and the metalloproteinases form some kind of non-covalent multimers, and if those are also surviving I think it's a strong vortexing that you make. So if they are still in this complex shape after you have them once or twice encapsulated.
Yeah, definitely. So one cool thing about our nanoparticle system that I didn't mention is that it suffers a auto hydrolysis process, meaning that the nanoparticle eventually will start auto hydrolyzing its ester bonds, making it like it's not enhance, but yeah, like enhancing its own size. That will actually form some water tunnels within the polymeric matrix, which is the thing that allows these toxin families to travel outside the matrix. So it's super interesting, and it would be super interesting to evaluate if there's an interaction between these toxin families, to see if they are like leaving the matrix, forming these complexes, or if they're living by themselves. Definitely, yeah.
There's one question in the chat. In long term, would you be planning to encapsulate individual venom components, so single toxins, into nanoparticles and test their efficiency?
Yeah, of course. So I was actually hoping for someone to ask this question. If you notice, we were encapsulating whole venom extract. If you go to a pharmaceutical company and you tell them, or you try to sell them this type of idea, they will, it's a Mexican slang, they will slap you in the face and pretty much tell you to go off with it. Encapsulating or using a whole venom extract as diverse as the whole venom, it's not profitable, and it has so many layers of things that could go wrong for a patient or in a clinical trial, that it's not suitable to develop something. So what we were trying to do is to put like the first steps in order to later on isolate and purify these toxin families, in order to encapsulate isolated and purified toxins, and then evaluate them with in vitro analysis, and later on in vivo analysis. So that's like the next step. And we're actually about to receive a batch of isolated toxins from another lab in central Mexico. And that's pretty much what we're hoping to do within the next 2 to 3 months. So yeah, great question.
Thanks. And the last question from the chat is about the specificity. How would you render the venom encapsulated nanoparticle specific to different cancer cell types?
Oh, so I didn't actually put that graph also, because I didn't want to. There's so much information, right? So we actually evaluated the cytotoxicity of these nanoparticles in different tumoral cell lines, and we were able to determine which tumoral cell lines were more prone, or were more, what's the word in English? Which were more suitable to evaluate these type of nanoparticles. And we determined that breast cancer was amongst them. So by itself, it's hard to determine by just using this nanoparticle which cancer type will be the most suitable to be evaluated. You have to consider stuff like which receptors are, for example, more expressed in certain types of cancer cells, which will be more suitable depending on the material that you use to encapsulate. So what we oversaw, let's say it like that, with this nanoparticle system, is that generally speaking, and in a very general manner, most of solid tumor cells are highly permeated towards nanoparticles sub 4 nanometers in diameter size. In order to consider which tumoral cell line will be more optimal or suitable for a specific type of nanoparticle, you would have to consider the nanoparticle material, and if you would like to put something on the surface of that nanoparticle, let's say an antigen or an antibody, in order to actively deliver it to a specific cancer cell line.